Okay. Hello, everyone. My name is Samir. I'm a fourth-year PhD student in biomedical engineering here at UW-Madison, and it's my honor to welcome Dr. Luke, a distinguished chemist, and as he humorously describes himself on Twitter, a fairy godmother to many imaging labs. His career has been defined by pioneering work in the field of fluorescent dye technology. Native of rural Southern Oregon, Dr. Luke earned his BS in chemistry from Oregon State University, where he gained early research experience in synthetic organic chemistry. After 4 years of After 4 years in industrial R&D, he pursued his PhD at the University of Wisconsin-Madison, developing novel fluorescent dyes to image biomolecular trafficking. Now at Janelia Research Campus, Dr. Luke focuses on creating advanced fluorogenic tools for high-resolution imaging, revolutionizing neurobiological research. We look forward to hearing his insights into these innovations today.
Wonderful, thank you. All right, so I want to really thank the organizers. It's been just special to be back in Madison. I haven't been back for like 5 years, but since before the pandemic, and it's been great to see a lot of friends and users in this meeting. So my good friend and mentor and former boss, Jerry Rubin, often says, you know, I've never seen a talk with too little data. And so I'm going to test that today. And so I have a little bit of data, the latest stuff from the lab, but I'm going to spend a little bit of time at the beginning, sort of telling a story to put our work in context. You know, where did all these dyes come from? And then at the end, Kevin asked me to talk a little bit about our efforts in sharing the dyes that we make. And so I'll talk about what we've done and also what we're planning to do. And I'd love feedback from this audience on what we're thinking about. All right, so about 17 or 18 years ago, across the street, I drew this plot. And it shows 30 commonly used fluorescent dyes in biology. They're graphed according to their brightness versus the absorption max. That's what each one of these points denote. And then the corresponding structures are colored according to the emission maximum of the dye. And the point of this slide is not to go through every structure, but it's just to showcase that we have a large number of fluorophores that span the UV, the visible, and the near infrared. And as organic chemists, we can take these molecules and tune and tweak them for specific biological applications. And so that's what my lab does. And I'll talk about that a little bit later. But because I'm an organic chemist, and I know you guys don't want to hear like an hour of organic chemistry, I'm going to start with answering this question. Where did we get these dyes? Because it's a fun story, and it puts some of our work in context. So really, the development of small molecule fluorophores, the discovery of fluorescence, and the development of instrumentation to measure fluorescence all begins with this one compound, quinine. Quinine is famous because it's an anti-malarial agent. It's a natural product from the bark of chinchona trees. It kills these plasmodium parasites that feed on our red blood cells and are transmitted by mosquitoes. And so a while ago, people realized we should put this stuff in the water, and that's what they did. And still, tonic water has quinine in it. It's the nice bitter flavor in our gin and tonics. And this is more of a chemistry joke, but Fever Tree, their tagline is made with natural quinine. And as if we have this huge store of synthetic quinine that no one wants. I mean, probably only made like a couple milligrams of synthetic quinine in history of the world. But this is a thing. Quinine was actually the first small molecule fluorophore observed, and the person credited with this observation was John Herschel. Herschel was a polymath. He did a lot of different things. And he wrote this paper in 1845. And I like reading the title of this paper because we don't write titles like this anymore. on a case of superficial color presented by a homogeneous liquid internally colorless. And in this paper, he goes on and on and on about a single experiment. He bought quinine sulfate. It was a drug. You could purchase it. He dissolved that in water. He took that vessel, he put it in the sun, and he noticed this weird blue tinge coming off the surface of the solution. And so he wrote about what this possibly could be and then moved on to other things. And this observation then percolated in the literature for a while. And a few years later, George Stokes wrote this seminal paper on the change of refrangibility of light in 1852. And in this, he showed that this weird blue tinge coming off the surface of the solution wasn't some weird scattering. It was actually absorption of one wavelength and then emission of another longer wavelength of light. If we were going to translate refrangibility into modern terminology, this would be the change of wavelength or the change of color of light, but this is before we knew light had wavelengths. And so realizing he had discovered a new phenomenon, said I can give it a name. And this was the name that he chose, dispersive reflection. And if this name had stuck, I'd be up here talking about dispersive reflexiphores, and all our acronyms would be different. But no one liked this name, including Stokes. So in a footnote on the same page, he says, I confess, I do not like this term. I'm almost inclined to coin a word and call the appearance fluorescence. And so that's the name that stuck. And this is the legendary Stokes experiment. It's not really unclear if he did this. It's not in the paper, but it said that Stokes, this is my daughter's drawing of Stokes, she's 8, went into a church one day with a solution of quinine in a clear glass bottle, and then a goblet of white wine. And so he used a blue stained glass window as the excitation filter, and then the wine as the emission filter. And so if he observed this light through the wine, he would see nothing, it would all be absorbed, but he could see this fluorescence emission from the solution of quinine. And then later, which is actually published in his paper, he repeated this with colored glasses and other solutions used as filter sets. All right, so quinine was the first small molecule fluorophore, led to the discovery of fluorescence. Gotta get used to this thing. Now, around the same time, the Industrial Revolution was in full swing. And one of the byproducts of this revolution was a substance called coal tar. This is the gunk left over after you pyrolyze coal to make coal gas to light the street lamps in London or to make steel. It's this crazy mixture of a bunch of organic molecules. And for a while, chemists just had fun distilling these things out. Many of them had interesting smells. They called them aromatic. We still use the term aromatic to describe phenol systems and other similar things. But eventually chemists thought, this is getting boring. Maybe we could take the stuff that we're distilling from coal tar and then do chemistry on it to make new molecules. And a major proponent of this idea was August von Hofmann. He was the president of the Royal College of Chemistry in London. He's the guy that coined the term organic synthesis. And he had a student in his lab, William Perkin. He was 17 or 18 at the time. This is a picture when he was a bit older. And together they cooked up this idea, what if we could make quinine in the lab? Rather than having it grown in trees half a world away, what if we could just take coal tar and magically make quinine? So that's what they set out to do. And this was their proposed synthesis. And I know this audience isn't really mostly chemists, but if you can think back to sophomore organic chemistry, you might realize this is not going to happen. This is N-L-toluidine, which they can make pretty easily. And for some reason, they thought, oh, we'll just make quinine out of this if we oxidize it. Remember, this was before all our nice spectroscopy techniques. All they really had at their disposal was elemental analysis and some simple functional group tests. And so from that standpoint, it kind of makes sense. You can take 2 molecules of this, add some oxygen, and you'll get the chemical formula for quinine plus a water molecule. So they tried this, it failed. Van Hofmann said, forget it, let's not work on this anymore. Perkin, for some reason, thought this would be a really great thing to keep trying, just oxidizing aniline molecules. He had built a lab in his parents' attic. So he went home, he distilled out aniline. It was contaminated with these toluidines. He oxidized it, and as the legend goes, he's cleaning up, and one of the solutions turned purple, and he had accidentally made the first synthetic colored compound. He later called it movine. And the fact that it was purple was a big deal. At the time, all colored compounds came from natural sources, and purple was the hardest to get. So the best purple dye was this Tyrian purple. That's a natural product from these spiny sea snails, and you have to get a lot of these snails to get a little bit of this dye. And so purple had always been associated with riches and royalty, but now Perkin can make this really great purple dye from super cheap starting materials. So being smart, he promptly dropped out of graduate school, filed a patent, started a company, and that was the birth of modern chemical industry. So many of the companies that we associate with pharmaceuticals or other chemicals started out making synthetic dyes in the mid-19th century. And a lot of the fluorophores that we still use today were developed during this time. So fluorescein was synthesized in 1871 by Adolf von Baeyer. Fluorescein has really played an outsized role in fluorescence imaging. So fluorescein diacetate was an early molecule that you could load into living cells. And then people figured out it was pH sensitive, and so you could actually use it to report the cellular environment. It's really the first live cell indicator. And then for this audience, many of us know about FTSE, fluorescein S-thiocyanate, the first shelf-stable amine reactive label for antibodies, and this really drove the development of immunofluorescence. And then the rhodamines pop up in the patent literature a few years later, synthesized by this Swiss guy, Maurice Ceresol, who was working at BASF. And again, dyes that we still talk about today are in this original rhodamine patent. Tetramethylrhodamine, I'll talk about this a little bit later. Rhodamine 110, the basis for many of the green dyes that we use, like Alexa 448 and rhodamine B, were all described in this original report. One little thing that I think is fun to look at is how dye chemistry then drove early pharmaceuticals. And this includes dyes going to analgesics. And the story here really begins with companies making dyes. It's a little bit hard to see on this screen. Maybe you can read the company here. This is a company at the time was selling dyes and apparently showing how they would dye feathers to make hats and other things. I like this page of the catalog because it has some rhodamine dyes on it. But the company is Bayer, which we don't think about as a dye company anymore. So at the time, Bayer was making this blue dye, benzoazurine G, and it was competing with natural indigo, which was controlled by the British. And to make benzoazurine G, they were taking phenol, they were nitrating it, you get two isomers. They were taking the orthonitrophenol and making benzoazurine G. And they would take the other isomer that you get from this reaction, paranitrophenol, and they were just storing it in barrels in the field next to the factory. So someone at Bayer said, we got to figure out something to do with this crap. And so some enterprising chemist said, oh, well, take this, we can make this molecule. And they found this was an efficient pain reliever called phenacetin. Now, the problem is phenacetin can be metabolized into two things. This is actually a prodrug. One is 4-ethoxyaniline. This causes kidney failure. This is actually probably what killed Howard Hughes. The other thing is acetaminophen. So we still use this today. This is Tylenol. So basically, Tylenol came from a byproduct when the Germans were trying to take over the blue dye industry from the British. So that's kind of fun. Bayer also developed the first antibiotic, and that was screening their dye library and finding this compound Prontosil, which was the first sulfa drug. It's really a prodrug. It gets reduced inside cells. And the guy that did this was Gerhard Domnack. And this is a great portrait of him by Otto Dix. And you can see this colored test tube, which is this compound here, and won the Nobel Prize in 1939. All right, and just to close out this story, quinine would rear its head one more time in the development of instrumentation to measure fluorescence. And that was during the Second World War when the US malaria program was started. And the idea was to replace quinine. Most of the quinine, the cinchona tree plantations were controlled by Imperial Japan. So they wanted to replace this with synthetic drugs. And they realized that this quinoline moiety was both the fluorophore and the pharmacophore. So they thought, oh, we'll make a bunch of new derivatives, and then we'll use fluorescence to see if they get to the blood where the parasites are. And so that was the idea. And this led to the improved dosing of this compound, atabrine, and the rediscovery of chloroquine, which now everyone knows about because of COVID, and especially its hydroxy derivative. And this was a partnership between the government and industry. And so before World War II, fluorometers were really rare, but afterwards they were commercialized. And now I'm sure everyone has a fluorometer in their lab in one way or another. So this is just an early thing, a new analytical instrument, this advertisement, spectrophotofluorometer. All right, so that's where we get all these dyes. And in our lab, we work on these classic fluorophores, and that includes fluorescein and the rhodamines. And we like these dyes because they're near the top in terms of brightness and span a good chunk of the visible region of the spectrum. And in particular, we really like rhodamine dyes because they're flexible scaffolds for building a wide variety of fluorescent probes. So one thing you can do very easily is tune their spectra. So this is that simple rhodamine, rhodamine 110. So it's like GFP or fluorescein, blue excitation, green emission. If you just alkylate the nitrogens, you shift things 50 nanometers into the red. If you tie back those substituents, you can get another 25 nanometers into the red. And then a more modern way to tune the spectral properties is to replace this oxygen with a carbon or a silicon, and that will get you 60 or 100 nanometer shifts into the red respectively. You can also make fluorogenic molecules, molecules that turn on in response to some event. So here's an example. This is a protease substrate, so you can acylate these nitrogens, locks the molecule into this non-fluorescent lactone form, but removal of these acyl groups through the catalytic action of a protease here can recapitulate the fluorescent structure, giving you a large increase in both absorption and fluorescence. And so we think that it's this interplay between this lipophilic lactone form and this fluorescent zwitterion form, we call the equilibrium constant KLZ for lactone zwitterion equilibrium, that really controls how dyes behave. And so, so you can have dyes that have relatively low KLZs. They remain quiescent until they bind something that shifts the equilibrium and turns them on. So you can make fluorogenic molecules. And then you can also have dyes that are mostly in the zwitterionic form, but have some propensity to close up. And these often show high bioavailability. And our model is that they can slip into lipid bilayers and come out the other side. And I'll give a few examples of how we can make dyes that are highly bioavailable and then use them in intact animals. All right, so those are the wonderful things about rhodamine dyes, but the problem is that the chemistry really sucks. And I'll just show you one example of this, the simplest rhodamine, rhodamine 110. So this is the original patent, the experimental from that 1887. Here's the first report in the primary scientific literature. There's this wonderful series of Soviet chemistry papers reinvestigating rhodamines in the 60s. And then here's a paper that's more modern. And basically, all of these are doing the same thing. So you take aminophenol and phthalic anhydride, and you condense them together with sulfuric acid at fairly high temperature, 180 degrees centigrade. So that's all done, whether it's 1887 or 2013. The only difference here is there's a handle for bioconjugation included here, so you can attach this molecule to some other thing. So again, across the street, I used to do this chemistry. Then I went home to Oregon, and my mother-in-law would have to fix all my jeans because I had tons of holes in all my clothes from working with Schwafelsour, concentrated sulfuric acid. So when I started my lab, I wanted to drag dye chemistry from the 19th century, maybe to the 20th century, or even the 21st century. And so over the years, we've developed a large number of chemical methods to make rhodamines and fluoresceins. So we've used reduced or leucorhodamines as synthetic intermediates. We've used modern reactions like palladium catalyzed cross couplings to transform fluoresceins into rhodamines. We've made new fluoresceins by using modern renditions of classic reactions like these turbo-Grignard. Unfortunately, this chemistry doesn't allow you to make rhodamines directly. So you metallate here, you add into this electron-rich system. These are nitrogens, this doesn't work. So more recently, we've come up with other ways to make rhodamine dyes directly, where we can make these dibromides and then metallate them and click them onto acids or anhydrides. And so the nice thing about this is this chemistry allows us to diversify up here on the top of the molecule. And this chemistry allows us to diversify on the bottom of the molecule. So now with these methods, we can make pretty much any rhodamine we want. And the question is, if we can make any rhodamine, which rhodamine should we make? And the Janelia fluor dye story really begins with almost a throwaway collaboration with Bob Tejan, who was president of HHMI and had a small lab at Janelia. And so Tej came into my office one day and said, I want to do this experiment. This isn't in cells. This is just in vitro single molecule imaging. He built this stabilized turf system with Steve Chu. And he wanted to watch RNA polymerase interact with DNA fixed to a cover slip. And they had a really slick way of reading out the transcript with a green dye. And so they wanted to label the RNAP with a red dye. And our idea to label RNAP was to use the halo tag. So you express your protein as a fusion with this halo tag developed here in town at Promega. The halo tag reacts with a ligand that you can attach to a small molecule fluorophore. And if you just mix these two things together, you get a fluorescent bioconjugate where the fluorophore position and stoichiometry is well defined. And so Tej had a very simple request. He said, can you make Psi5 halo tag ligand? At the time, you couldn't buy any good far red fluorophores suitable for single molecule imaging with this halo tag ligand attached. So we actually had Psi5 and the halo tag ligand in the freezer, and Tej was president, so we did it that same day. But we purified it, but this was kind of early in my career. I didn't want to make things seem too easy, so we waited a couple weeks, and then I emailed Tej, said, I think we finally made it. And we published the science paper with them. But that really began our interest in developing dyes for single molecule imaging, and Tej and many others were starting to do single molecule imaging, not just in vitro, but also in live cells. Again, using this halo tag to attach bright synthetic fluorophores onto proteins and then watch them move around. This was at a time when many cool microscopes were being developed at Janelia and elsewhere. This is the multi-focus microscope developed by Mats Gustafsson. So it images in 9 focal planes simultaneously, so you can watch individual molecules moving around in 3D. But again, the issue was chemistry. So the only dye with the requisite brightness, photostability, and cell permeability for these sorts of experiments was this classic fluorophore tetramethylrhodamine. This is in that original patent from 1887. So it's a decent dye, it gets into cells, it labels the halo tag rapidly, but it has a low quantum yield, 41%, and modest photostability. You might think, well, use a more modern fluorophore, like one of the Alexafluor dyes. And I love the Alexafluor dyes. I worked on them when I was at Molecular Probes. But Alexafluor dyes weren't designed for live cell experiments. They were designed to be antibody labels. And so they're basically sulfonated laser dyes. And this is Alexafluor 546. And so they have a bunch of substituents to tune wavelengths to improve photostability and to improve water solubility. So these are brighter, but they don't get into cells very well. So our idea was to make a dye that would have the brightness and photostability of an Alexafluor, but the small size and cell permeability of these classic dyes. So that's what we set out to do. And we thought a little bit about photophysics. How do we make brighter rhodamines? So very basic. You have tetramethylrhodamine in the ground state. It absorbs a photon, gives an excited state, and then the molecule can relax back down to the ground state through emission of another longer wavelength photon. But this only happens 41% of the time. What about the other 59% of excited states? So these dyes are particularly susceptible to a mode of non-radiative decay. It's called twisted internal charge transfer, where you get electron transfer from this nitrogen into the xanthine system, and then a twist around this carbon-nitrogen bond. This then relaxes back down to the ground state without emitting a photon, so it competes with fluorescence. You can also get intersystem crossing 1 or 2% of the time to form the triplet. This then can relax back down to the ground state through sensitization of singlet oxygen. And then this singlet oxygen can do bad things. It can pull off an electron from this nitrogen. You can get loss of a proton and then ultimately dealkylation. And that seems to be a first step in a photobleaching process. So we thought if we could slow down this, we could improve brightness. If we could slow down this, we could improve photostability. And we realized that both of these mechanisms involve pulling off an electron from this nitrogen. So we thought, let's just make that harder, and we can make a better dye. And our very simple idea was we add a carbon atom on either side, tie this back into an azetidine, and azetidines have really high ionization potentials. It's really hard to pull off an electron. Maybe we could make a better fluorophore. Now, no one had done this because the chemistry wasn't there. But fortunately, we had developed this method to take fluoresceins and turn them into rhodamines using this palladium catalyzed cross-coupling. It works for azetidines. So we could do this in good yield. And we made the first Janelia fluor dye, JF549, named for its absorption max in water. And we saw an improvement in quantum yield, more than twofold. And so this is a minor modification, just the addition of a couple carbon atoms, so it doesn't change the spectral properties. There's really a shift of just 1 nanometer, so it preserves what we like about tetramethylrhodamine. But in live cell single molecule imaging experiments, it was twice as bright and lasts twice as long versus tetramethylrhodamine. So you got 4-fold the number of photons out. And this has enabled now a large number of live cell single molecule imaging experiments. This is one of my favorites from my colleague James Liu. So the little magenta fireflies are wild type Huntington protein whizzing around, labeled with JF549 using the halo tag system. And then there's a mutant Huntington protein labeled with GFP. So that aggregates, but you can also see that the diffusion of the wild type protein slows down in the presence of that aggregate. All right, and so from that one discovery, where we took this classic dye, added a couple carbon atoms and made JF549, over the years, we've made a number of derivatives of these things. So we've made photoactivatable derivatives. We've tuned the spectra across a pretty broad range. We've made fluorogenic molecules, molecules that turn on when they bind the halo tag or other biomolecular targets. We've made bioavailable fluorophores that work in animals. We've made photosensitizers, which sort of languished for the last 4 years, but there's renewed interest in these due to photo proximity labeling technologies. So there's a recent preprint from Carrie-Ann Backus at UCLA using JF570, this dye here, to do proximity labeling. We've deuterated dyes to further improve them. We've added stuff to the dyes to make sensors or other things. And then we've made spontaneously blinking fluorophores. I'm just going to talk briefly about some deuterated stuff, some spontaneously blinking stuff, and then a few other examples of using fluorogenic or bioavailable fluorophores, just to give you a flavor of what we're working on right now. All right, so one of the things we can do is incorporate some of these bioavailable or fluorogenic molecules into sensors. So examples of this include Voltron. This is a voltage sensor that we developed with Eric Schreider. It's a voltage sensitive opsin fused with a halo tag labeled with a dye. When the voltage when the membrane depolarizes, the absorption of the opsin goes up. You get an increase in FRET and a modulation in your fluorescent signal. A really good dye is this bioavailable JF608. It's long enough so you can do optogenetics, but short enough to get efficient FRET. And Adam Cohen, who's really the discoverer of this voltage-sensitive opsin, has been using this system to do very fast voltage imaging, watching action potentials propagate across a neuron. We can also use fluorogenic dyes to make sensors. So if you make a circular permuted halo tag, put a environmentally sensitive fluorophore in the middle of two sensor domains, like things that bind calcium. You can make sensors here. We're using these fluorogenic dyes like JF635. And these work pretty well in culture. These are neurons that are being electrically stimulated. And I think my movement from PowerPoint to Keynote made the movies not loop. And then last week, Eric Schreider and his postdoc, Helen France, published an update on this, a system they called WALOCAM. The W is for this tryptophan that quenches fluorescence. So when calcium binds, you dequench the fluorophore, getting a large increase. And the nice thing about this system is it's universal. You don't have to use fluorogenic dyes. You can use any dyes, including highly bioavailable fluorophores like this JF669. And so here you can do whole brain imaging in zebrafish. And this is a far red sensor, so it can be imaged simultaneously with, with standard green and red sensors like GFP and red fluorescent protein-based indicators. All right. We've also tried to improve the fluorophores through deuteration. So if we go back to this photophysical scheme, our original idea was to slow down the photobleaching process. So there's the loss of a proton here. We make this carbon-hydrogen bond stronger. Maybe we could make a dye that's more resistant to photobleaching. When we started digging into the literature, so the hypothesis is if we deuterate this, we could make a dye that's more photostable. When we started digging into the literature, it turns out there's a pretty pronounced secondary isotope effect on the ionization of means when you deuterate them. So that led us to refine our hypothesis. Maybe we can improve both brightness and photostability just by deuterating these N-alkyl groups. And that works. So we can deuterate the azetidine or even deuterate the 5-membered ring. And in some cases, the 5-membered ring is actually the winner. So this is a dye called JFX650. This is just imaging in yeast by Ben Glick's lab. And this JFX650 is the last man standing. And we've used this deuteration trick to make very good dyes for in vivo use. So this is JF669. It works really well if you just inject 100 nanomoles of this dye in vivo. It will homogeneously label halo tag expressing neurons in the brain. If we deuterate it to make this JFX673, it's a little bit brighter, more photostable. And so with this, we can then do pulse chase labeling in vivo, where we have a mouse expressing here the synaptic protein PSD95, so fusion with the halo tag. So we pulse with this ligand. We then do something to the mice. We chase with this other color, and then we dissect an image. And so from this, we can get data where here's the pulse signal, or here's the pulse, here's the chase. And then from this, we can calculate protein lifetime. And we can do this across the brain. So it's only one protein, but you get the whole brain, and it's post hoc. And this is a mouse where they experienced an enriched environment during this time period. And you can see sort of interesting things. There's a gradient in the cortex from long to short lifetime, and there's some differences in the hippocampus. And so this is a nice way perhaps to figure out where to aim your 2-photon microscope if you want to do functional imaging or follow up with more comprehensive mass spec. And then finally, I'll just touch briefly on these spontaneously blinking fluorophores. So these have been around for about a decade now. They're hydroxymethyl derivatives of silicon rhodamines, and they work by spontaneous protonation to form a fluorescent species. and then they go back. And the dye that's been around for a long time is HM-SIR. And the problem with HM-SIR is that the duty cycle is too high. So too many dyes are on for standard localization microscopy or other similar imaging. And so this is just a nucleus of a cell expressing halo tag histone H2B. And you can see a little bit of linking, but you can't do localization microscopy here. So we made a whole series of these dyes incorporating the azetidine and also some other functionality with the goal of improving the brightness and also changing the duty cycle. And so this is what we get, all same cells. And you can see that we have a decent gradient going from high to low. And so you have dyes that have a fairly high duty cycle, just high, low, and very low. And it turns out these dyes work pretty well for localization microscopy. This is with less ligand. So this is JF630B, B for blinking. And so you can do nice localization microscopy with the halo tag, no buffers. This is just blinking in PBS. So here, Tom20, Sec61 beta for the ER or the nucleus. You can push this and do live. Of course, with live SMLM, the frame rate is pretty poor. And so one thing we've been investigating, this is early days, but using these dyes for live cell SOFI, I think this is the first example of using a blinking small molecule fluorophore for SOFI in a live cell. But it works pretty well, and you can get maybe 1 Hertz type imaging. You make each SOFI image from 100 frames. All right, so what's next? We're making dyes that are further redshifted. we're like trying to put deuterium in everything. I like this. This experiment's fun because it's like only a Janelia experiment. So the starting material to make this was a $20,000 reaction. Of course, we did it without the deuterium first to make sure it worked, but it'd be hard to do that in a standard academic environment. We've made cell-permeable fret quenchers. So you can do fret flim type things using the halo tag and snap tag. These work actually quite well, both as the halo tag and the snap tag. We're making bioavailable photosensitizers for either killing cells like this in the fish or for this photoproximity. We've made impermeable but fluorogenic molecules, so you can bind just the surface proteins. And then I want to take just a couple minutes and talk about this other effort called open chemistry. All right, so I'm basically a poor hippie from Oregon who was given a remarkable opportunity at Janelia, and one of the things that we wanted to do is try and give away as much stuff as possible. And just to preface this, I want to kind of go back to the 90s, where maybe some of you remember what science was like back then. I really miss the 90s. But if you wanted to label something, you would use an Alexa fluor or one of these stains. If you wanted to watch cellular changes, you would use Flow4 or BCECF. If you wanted to screw with your cells, you'd use a pharmacological agent. And if you wanted to activate something with light, you'd use a cage compound. And one of the companies that was really helpful during that time was Molecular Probes. They had this huge catalog. Dick Hogland, the founder, his mantra was, if we're making money on a product, we'll keep selling it, which is a terrible business decision, but it really benefited the community. All right, so then in the aughts, when I started my lab, genetically encoded tools were all the rage, fluorescent proteins. You had indicators. You just knock out your protein Y, do a pharmacological manipulation, and then, of course, optogenetics happened. And, AdGene, which popped up, sharing plasmids, is a major reason why everyone uses genetically encoded tools. They're also better in many cases. But now what I just told you, and many other labs are working on this, these hybrid tools which have small molecule components and protein components. So these tagging systems that we love, these indicators that we and others have developed. There's cell type specific pharmacology, where you can get both molecular and cellular specificity. And then there's this field of photopharmacology, where you can get specific manipulations of proteins by putting photoswitchable drugs on the surface of cells. And so this begs the question, do we need an AdChem to sort of complement AdGene? And we've thought about this, and we've sort of moved towards it. And so I'll just start with the things that we had. We had really efficient chemistry to make these molecules. We then had used that to develop a bunch of useful compounds. And we also had support from our organization. So Genelia really exists to build tools and give them away. I had hired this guy, John Grimm, that I knew in graduate school here, best bench chemist in the world, had gone to Merck, and I was able to entice him away from Merck and come join Genelia. And then I also had my boss, Jerry Rubin, and Jerry loves giving stuff away. He famously, when he figured out how to make transgenic flies, he showed up at a meeting, presented the work, and then said, by the way, I've prepared kits for every single one of you in the audience, so you can do this at home. And he handed those out after his talk. So, it's sort of in our DNA. So what did we do? So we decided to give away everything for free to everyone. That means everyone, even our competitors in the chemistry space. To do this, we have hired typically 1 or 2 technicians. These are former members, and they spend a year or 2 in the lab and then go off to grad school. So Anna went to Caltech, Anthony went to Harvard, Natalie went to UC Irvine, and Katie went to UCSF. We decided to only require a citation. Most chemists ask for authorship, sometimes like co-corresponding authorship, and we didn't want to do that. And then it's got to be a pain, so we developed this charmingly clunky website called dies.genelia.org, and this was done primarily by Anastasia and Megan. We have support to pick up the stuff and package it and send it to all of you. And then if you register here, Tim Brown is sort of the outside person that validates that you're at a nonprofit institution. So I'm not the gatekeeper and saying no to the people that have said bad things about me in talks or whatever. And so these are numbers I got this morning. So we've sent out about 5,000 orders, a lot of aliquots to a lot of labs in many countries. So this would be, if you believe Promega's current pricing for the Genelia floor dyes is like, $230 million or something. So it's a lot of compound. And it's really just one freezer. This is a tweet from a few years ago from John. And we just have this freezer, and orders come in, you get a pick list, the technicians fill them. We have an inventory system, and we just ship things out every day. All right, so what have we learned? So we can't do everything, but neither can companies. So we don't give away everything that we make. We give away the things that we can give away. Larger compounds, like when we're trying to sell it to chemists, building blocks, those sorts of things, we've let Talkris and others handle those. But for halo tags, where we can give away a very small amount and it can last a cell biology lab, a year, that's something we can do. I think it's important to recognize, and I know folks here work for industry, companies exist to create value and make money. They're not just a service thing. Probes was an anomaly, and sometimes even if you have a good tool, there's an issue with market size or even the business model of the company that might own the IP that can limit commercial availability. So I've also learned that fun things happen when reagents aren't scarce. So, early in my career, Winfried Denk used to hang out a lot at Genelia, and he would just come into my office and grill me for hours about what we were doing. And, he would always say, There's not enough goofing off in science. And so I'm a firm believer that, we want to give away reagents so graduate students don't feel like, oh, I've got to reserve this for my, in case a reviewer asks me to do this experiment, right? We'd rather just have people be creative and be able to try new things. And so a great example of this are some new indicators and integrators. So during the pandemic, we sent a ton of stuff to Adam Cohen. Someone in his lab had a great idea for a new integrator system, this ticker tape system that he published a few years ago. And, it wouldn't have happened unless there was a box with a ton of our dyes just sitting there. And he realizing that we could use them and there would always be more. And then removing authorship is really good for science. So I know all of us know this, but most of the time it's negotiated by PIs, not students. And I would get emails from students CCing their PI, sort of saying, hey, I saw your paper, and we'd love to use the thing, and let's talk about the collaboration or the authorship thing, and they just want to get it out of the way. And then I just tell them, yeah, we don't do that here. So, you're free to use our stuff, just cite our paper, or acknowledge us if you want. That has really, I think, accelerated science, because suddenly this poor second-year graduate student doesn't have to worry about some dude in Virginia, going to gum up their author list. And it's really facilitated open communication, because there's not that underlying weirdness about authorship in the emails, right? We can just talk about science because it's sort of a foregone conclusion. All right, so what are we doing? We've persisted but evolved. We've hired new technicians. So we have Sarah and Carla. Sarah, unfortunately, will leave us to go to graduate school. She's applying right now. We've also had to adapt as molecules have been commercialized by Promega and others. And so what do we do in that situation? Do we not give them away? Do we only give them away to select people? And that's been difficult. We've also learned that we need a separate nonprofit. We have a pretty good logistical foundation for distribution, but it turns out that if you're part of an organization like Hughes with really deep pockets, they don't want to bring in other stuff, and there's liability. So we're going to have to create a separate entity. And then right now we're really debating the best way to set up AdChem. So there are different models. There's the grinder model, as I call it, where you just have like a social media thing connecting a tool builder and a tool user. I want to call it builder, but no one's really caught on to that. There's also Etsy, where you have a marketplace where you buy, but then it's shipped directly from the lab. And then there's the Amazon, where everything is centralized. And that's kind of what we've been doing. But this is sort of what we're thinking about. And then, we're trying to partner with other entities, perhaps AdGene. CZI is interested in funding some of this, the Brain Initiative, et cetera. But I'd love to hear any feedback or ideas that you might have. All right, so with that, I'll end. So here's the lab. Here's open chemistry. We work with a lot of people inside Janelia, outside. And I just really want to mention John Grimm, who has been the person driving a lot of the Janelia floor stuff at the bench, developing the chemistry and all the molecules. So thank you very much. Happy to answer any questions. Science or otherwise, yeah.
Yeah, I'd love to kick off the question round. It was a lovely talk, and I'm sure many more questions will follow. You mentioned the pulse chase experiment. I actually don't know how those work. So maybe if you could, explain how that works and how your dyes play a role there, I would be very thankful.
Right. Okay. I don't know how to do PowerPoint, so I'm gonna. Oh man, I have to go back. So basically, the idea is, so you have a mouse expressing the synaptic protein. Okay. Oh wow. Okay. In this case, PSD95. Oh yeah, perfect. You have this mouse expressing the halo tag fused to the synaptic protein PSD95. when you label this protein with the halo tag ligand, that's stable, it's irreversible, it's covalent, and so you pulse and saturate all existing proteins. You then wait some time, new protein is produced, and then you, label that. Okay, I can't see anything. You label that with then the chase dye, and so the new protein, the nascent protein, will be one color. The original protein will be the red color here, and then you go to orange. And then from this ratio of signal, you can calculate some protein lifetime.
These one, and also the dyes that are only become photoactive when the snap tag binds.
These are actually just, they're not they're not fluorogenic, but they are highly bioavailable. So we administer this IV retroorbitally. It will cross the blood-brain barrier and label neurons in the brain. So we have excess dye that then is just excreted. And so, but we've done the experiments, these pulse chase experiments, one right after another, to show that this dye actually can saturate protein in the brain to, almost saturate. And so you get most of it labeled with the red, and then you look at the new protein with the yellow-orange.
Thank you.
This is a question from Zoom. But first of all, thank you for your wonderfully bright dyes. And in your opinion, what's the possibility of significantly increasing the fluorescence changes in response to the environment? So for example, like the membrane potential and the calcium concentration, can those get even brighter and better?
Mm-hmm. Yeah, we think the sensor systems that we've developed can get brighter. And one cool thing, because we're giving away a lot of these dyes, that many other sensor developers have sort of adopted some of these technologies. None of the new work is published, but I know, there's a glutamate sensor, a dopamine sensor that works in vivo. There's a new potassium sensor. So the hope is, if we're providing the ligands, then that makes the sensor community free to develop new types of sensors and hopefully improved versions. So, Ahmed, who developed Voltron, is now working on new Voltron type systems using, some of these fluorogenic dyes. And so, hopefully the community will solve that problem after we've made these crummy prototypes. So yeah, don't tell Eric I said that, but yeah.
So when I teach confocal, I always tell students that there's no free lunch. You have to pay sensitivity with resolution, speed with something else. Apparently, I was wrong. My question, my question is, when you design all this, developing all these wonderful dyes, how do you avoid the toxicity?
That's a great question. So we worry about this a lot, and this, we didn't solve this problem. Actually, Promega solved this problem. So the beauty of the halo tag system is that it's so fast, so efficient, that you can label with nanomolar amounts. So I'm sure our dyes are toxic, but we never reach that level because we're like this experiment, we're injecting just 100 nanomoles of compound, so 80 micrograms or so of the ligand. For an entire mouse, it will saturate the tissue. So we're dealing with quite small amounts, and so we haven't had to deal with any toxicity issues. Of course, we've looked for it at those concentrations. We don't see it. Even at micromolar concentrations, they seem fine. But that's how we get around it, and it's not our doing, it's Promega's doing.
It's a beautiful talk. I was curious about the community side of your talk. That as you think about the not-for-profit, how do your peers, your fellow developers feel? Do you think there's potential of having the pro developer community join you and having other developers perhaps put their probes into your not-for-profit?
Yeah, so we tried to do this, but as I sort of joked, HHMI then pulled the plug on bringing out in some outside things, and that's understandable. There's a lot of IP issues and other things, but in general, I think that this idea of molecular probes not the company, but just this field in chemical biology has really grown. There's a study section with probes in the name now. So it's a big thing. And, you know, one of the issues is how do you get someone else to use your stuff? And it's very hard, you know, I'm privileged at a place that will pay for technicians to help me do this. Not everyone has that. And so we think it could be useful. The probe community is on board. And the way that we sort of think about it, one way of having this work is, you know, if you make a cool probe and, you know, people are requesting it, then you could apply for like a supplement to, you know, if you're like a brain initiative grant or something, then have it custom made. Then you'd ship that material, basically deposit that material into AdChem, similar to how you deposit material in AdGene. And then AdChem wouldn't have to make that thing. They might have to maybe purify it or aliquot it and QC it and all that. But that then would lower the cost for AdChem, at least initially. It also mean that the probe depositor, the probe developer has to put some skin in the game. They can't just say, make all my stuff because it's amazing. And, you know, you know, they need to actually like put up or shut up. So, so I think the probe development community is on board. They would love to see some of their stuff get out there and become available, and not at the expense of, say, a postdoc who's trying to finish up their project.
I actually had a quick chemistry question. You mentioned how expensive the deuterated samples are, and also I really liked your point about moving away from like boiling sulfuric acid. So in terms of like, I noticed the palladium, it tends to be rare and expensive, and the butyllithiums are going to be like pyrophoric. So I know that's kind of the new part of the new wave of chemistry. Are you guys trying to move towards any of the quote unquote greener methods?
Yeah, it's a good question. So I will say, I probably shouldn't say this, but these are pre-pandemic numbers, but you know, a vial that we would send you guys 100 nanomol's, which is, you know, $10,000 per mega, costs us 62 cents to make. And that's labor, everything. It's probably, you know, 72 cents now or something. The deuterated stuff adds 10% to the cost. So it's like not that much money. We do have this crazy idea. My first postdoc is now at a primarily undergraduate institution. And, you know, there's a drive to get chemistry students involved in research early on. And, you know, most of those projects are, let's face it, not awesome, right? They're sort of contrived or whatever. So our idea is, you know, is the undergraduate community a untapped resource for making dyes. And so we have a project now to try and move dye synthesis into something that undergraduate institutions can do routinely. Purification, all these things are difficult. But if we can do that, then we could have people making, you know, like what if we had Alexa 448 free for everyone? You know, you just request 5 mgs at a time. It's made at an undergraduate institution, shipped through AdChem, and, you know, we could do those sorts of things. So we are thinking about simplifying the chemistry, not just for environmental or cost reasons, but also to try and make it accessible for undergraduates to perform. There's more questions. Sorry, I went a little late, but yeah.